All right, we'll get started here. If you're just joining us, feel free to drop your name into the chat. Let us know who you are, where you're coming from. I want to thank you all for being here for our MWEE Practitioners Webinar. My name's Elise Trelligan. I work for the NOAA Chesapeake Bay Office, along with some of my colleagues that are on here today, Krista and Bart. And we work to support environmental literacy across the Chesapeake Bay region. And as you know, this is a part of a regular MWEE Practitioner Webinar Series that we host on a monthly basis. We cover topics related to designing and implementing meaningful watershed educational experiences, as well as unique science content that can serve as the basis for those investigations within MWEEs. Mandy, if you want to flip forward to the next slide for me. As you can see, we have a couple more scheduled webinars for this winter. On December 1st, we're going to be doing one on opportunities for alignment around sustainable schools and MWEEs. And then on January 20th, we'll have one on the topic of green infrastructure. And then on the right-hand side, you can see the NOAA Environmental Science Training Center. We have a couple of virtual workshops that are in the queue for non-formal educators for this winter. There's some really great ones. Registration will be opening for the Green Workforce and the Environmental Data Workshops in early January. And all this information, including the webinars, are available on our website, NOAA Chesapeake Bay Office. So that's linked at the bottom there. These slides will be shared with you at the end as well. This presentation is being recorded, and always the presentations can be found on the Chesapeake Bay Program's YouTube page under the MWEE Practitioner playlist. I'm going to flip to the next one for me, Mandy. So we're all really accustomed to the virtual space at this point, so I'll be kind of brief in reminding us all to mute our mics during the presentation. Krista and Bart can also help if you're having a challenge there. We might be able to mute you from our end. Also, I see a lot of smiley faces out there. Glad to see you all on video, but you can shut the video off for this portion during the presentation. And if you choose to turn your video back on if you have any questions throughout. So we have a wonderful speaker today, Mandy Bromillo. She'll be talking about the importance of forage and sustainable and developing a sustainable and healthy Chesapeake Bay ecosystem, and some ideas for bringing those lessons and concepts into the classroom. So I encourage you to jot questions down as they come to you throughout the presentation. She's going to take a couple of pauses over the course of the presentation so that she can answer them as they come into your head. So please drop those into the chat. Bart and Krista will be helping to pitch those as we go. And with that, I think I'm going to turn it right over to Mandy so we can get to the good stuff.
Thanks, Elise. And I just want to thank you all as well for your interest in this topic and attending today's webinar. As Elise mentioned, my name is Mandy Bromillo, and I am a fisheries scientist at the NOAA Chesapeake Bay Office. And just to give you a little bit of background about me, I got my bachelor's degree in ecology and evolutionary biology at the University of Michigan. And I also did an honors thesis looking at white shark behavior around cage diving vessels in South Africa. Actually, the photo behind me is a photo that I took from a cage diving vessel one of the summers I was down there. And sort of the experiences that I had as an undergraduate really sparked my interest in ecosystem connectivity, conservation ecology, and ecological interactions, particularly predator-prey interactions. So after I graduated from Michigan, I went to the Virginia Institute of Marine Science to get my master's in marine science. And there I studied blue crab survival, specifically looking at juvenile blue crab predator-prey interactions and trying to identify key predators of juveniles in seagrass beds. Now, as a fisheries scientist at the NOAA Chesapeake Bay Office, I provide support for a lot of different fisheries projects that come into the office. But another major aspect of the work that I do now is coordinating various work groups in the Chesapeake Bay Program, including the invasive catfish work group, the Chesapeake Bay Stock Assessment Committee, which focuses on blue crab management in the bay, and finally the Forage Action Team. And the Forage Action Team and the work that they're doing is what this webinar will focus on today. So before I get into it, just want to give a brief outline of the presentation so you know what to expect and what we're going to talk about. First, I'll be defining forage and its importance in the ecosystem and also to humans. I'll then introduce the Chesapeake Bay Program's Forage Outcome, which sort of directs their conservation and restoration efforts related to forage. And then I'll introduce the concept of ecological indicators and how they can be used to track forage status, and then how those ecological indicators can be used to inform management, ideally both fisheries and habitat management. And finally, I will present some potential education resources that you can use either for formal or informal education, things like lesson plans you can conduct in the classroom, some potential data sources if you have interest in looking at analyses, and also some other ecological indicator resources that you can look at as well. So first off, what is forage? Forage are typically defined as the lower to intermediate trophic level species that feed on microorganisms in the ecosystem. In this instance, plankton, both zooplankton and phytoplankton. And forage are the primary food source for many predators in the ecosystem. This includes large predatory fishes in the Chesapeake Bay. You might think about striped bass or bluefish or weakfish, but they also feed marine birds like osprey, and also marine mammals, which maybe you're not familiar, but we actually get dolphins in the Chesapeake Bay, particularly in the Southern Bay in the summertime. And historically, forage has referred to these smaller schooling fishes, things like river herrings, such as the alewife, different species of shad, and maybe two of the most important forage fishes in the bay, the bay anchovy and menhaden. However, forage sort of extends beyond these small fishes. If we think about forage as the link between plankton and these other larger predators, you might realize that this includes other benthic invertebrates as well. Things like clams, shrimps, little crustaceans called amphipods and isopods, and also polychaetes, which are these little bristle worms. And there's also a species you might be more familiar with. Juvenile blue crabs are an important forage item as well. So all of these species provide prey for larger fishes and even blue crabs, adult blue crabs in the bay. In fact, cannibalism is a key component of blue crab population dynamics in the bay. Fun fact. But who really cares about forage? I guess the easiest way to think about this is thinking about who we consume directly as humans. So there are some species like anchovy or clams, sardines in some instances, that we eat directly. But a lot of the forage species that we talk about are not actually consumed directly by humans. If you think about menhaden, it's a really oily fish, not something that we would typically consume ourselves. So another way to think about the importance of forage fishes in the Chesapeake Bay is what we call a reduction fishery. And that's essentially where we take these smaller forage fishes that we don't consume directly as humans, maybe things like anchovy and menhaden, and they go through this whole sort of really complicated reduction process, and we produce fish meal and fish oil. And these products are really high in protein and other vitamins, and are therefore used in various other commercial industries. So for example, they might be used in pet food. So this little cutie is my newly adopted dog, Cusco. And if he were to eat some salmon flavored dog food, it would likely have fish meal as one of the primary ingredients in that food. Fish meal is also often used in livestock and agriculture feed or aquaculture feed. As I mentioned, fish meal is very high in protein, but it's also very calorically dense. So they're used in these feeds to really fatten up those species that we're growing for harvest. So like I mentioned, yeah, aquaculture, maybe for like salmon or something, and then livestock feed. So if you're raising cattle or chickens, you might feed them some sort of meal that includes fish meal as well. Maybe something you might be more familiar with yourself is fish oil. You can find fish oil in pretty much any grocery store or pharmacy, and a lot of people take this as a omega-3 fatty acid supplement. So with all these different commercial uses, the reduction fishery of forage fishes is worldwide a multi-billion-dollar industry. So there's a lot of economic value for these small fishes. In an ecological sense, forage species are also incredibly valuable and are often considered the cornerstone of a healthy ecosystem. So the title of my presentation today is Forage, the Building Blocks of the Bay. And that's because they really provide this critical link in the food web between plankton communities and those larger predators. They're essential for transferring that energy up the food web. And as I mentioned, they're feeding these ecologically and economically important predators that we also will have interest in as humans, things like striped bass and blue crabs. And because they're feeding these important species, we know that they're supporting these lucrative fisheries, both commercial and recreational. And I looked up some numbers for you guys to get a sense of just how much economic impact these forage fishes have. So the direct global commercial value of forage fishes annually is $5.5 billion. So that's how much money they contribute to the global economy simply through the reduction fishery and our direct consumption as humans. The indirect commercial value of forage fishes is over $11 billion worldwide. And so that means that they are so vital in supporting these other fishery species that we're interested in, things like striped bass and blue crab, that they're really driving those populations and therefore driving the economics of those fisheries as well. And that $11 billion value is only considering the commercial value, so the fishes that we're harvesting for commercial use. That doesn't include the added recreational value, maybe from angling or charter boats, ecotourism, things like that. So clearly there's both economic and ecological value of these fishes. And in fact, they're so critically important to the ecosystem that a bipartisan effort was actually passed in 2019 called the Forage Fish Conservation Act. And that essentially sort of laid down some rules about effectively managing the species and conserving them, both for their own value, but also for the value they provide to the other fisheries that we're interested in. And I also was saying that by providing sort of prey resources for some of these other species that we're interested in, their own intrinsic value, they're also important for marine bird populations such as the osprey. Okay, and forage species are also really important because they provide ecosystem services. And ecosystem services are positive benefits that are provided to people or the environment. So many forage species are what we call filter feeders, so that they sift out all the plankton from the environment. So things like planktivorous fishes like menhaden. You can also think about clams. Similar to oysters, they're siphoning the water column. And in that sense, they help maintain water quality and prevent the formation of algal blooms by reducing excess phytoplankton. And I have a quick video I wanted to share here. This will play. Okay.
This video was filmed on August 13th in Cape Cod Bay. The fish you are looking at right now are called menhaden. They're also known as pogies. They feed by filtering phytoplankton, zooplankton, and other organisms from the water. If you look very closely at this video, you can actually see the plankton.
So I really like this video. It sort of ends with that punchline of menhaden being considered the most important fish in the sea. It's not something we really think about when we think about forage fishes. They don't get enough credit, really. So yeah, as I mentioned, really important for water quality, vacuums of the sea. So what does it mean if these links disappear? What happens if we lose our forage populations? So if we lose forage, I guess the first thing, the most obvious thing, is that we would start to lose food for predators. And with a decrease in prey resources, it's very likely that we would start to see decreases in those fish populations. And with the decrease in those populations, as well as the forage fish, we might see a decrease in these important predators as well. Top predators, things like the osprey and dolphins that we see in the bay. So additionally, we would also see reduced fisheries harvest for those important predators that we're interested in, things like striped bass and blue crab. So we'd see reduced harvest, less fishing opportunities, and obviously we talked about the economic importance of those species. So with the loss of those forage fishes that are fueling those fisheries, we would see a huge economic loss. As I mentioned, this is a multi-billion-dollar industry worldwide and a multi-million-dollar industry across the bay. Another thing that we can think about is the ecological impacts. So we know that forage fishes are sort of the primary predators of the plankton communities. So if we lose those species, we might see more plankton-dominated ecosystem, which could lead to more algal blooms. And these algal blooms, through their microbial processes, they tend to eat up a lot of the oxygen in the environment, which would lead to more what we call hypoxic conditions. So insufficient oxygen in the environment. And that also affects the habitat of species or fish species that we're interested in, which can result in fish kills. So the loss of forage obviously would have a huge economic impact from a fisheries standpoint, but it can also disrupt the balance of the entire ecosystem. So they're super important. And I'm going to stop here before we move on to the next section. If anyone has any questions, I guess Elise or Art or Krista, if you want to let me know what questions we have coming in, if any, at this point.
Mandy, no questions in the chat yet, but I'll give people a moment to drop any in. One thing that came to my mind, and you're starting to talk about the sort of effects that could potentially be seen. Are there any case studies that you're aware of, or like any examples of when this has occurred locally in the Chesapeake Bay?
Like severe losses of forage, you mean?
Yeah. Mm-hmm.
It's a good question. I don't know of any case studies specifically, like for any specific species that was reduced. We didn't see impacts on it. Yeah, I don't know. That's a good question. But I'll talk a little bit later too about how prey, like with shifting prey populations, predators will also shift what they're eating. So they sort of compensate sometimes in that sense. So, I mean, even if it would have to, I'm talking here like sort of hyperbolically, if we lost all of the forage in the bay, we would see these really significant changes. But if we do see changes in like one population declining, it's very likely that predators will try and compensate by feeding on something else that's more widely available. But that can also have other impacts with the other connections that are in the food web as well.
Gotcha. I don't see any questions coming in the chat, but Erin actually has one here. What threats do forage species in the bay face?
Mm-hmm. Yeah, so I guess the biggest thing, well, one of the biggest things for forage fishes anyway, things like menhaden in particular, is overfishing for the reduction fishery. So that is like catching or harvesting menhaden to be produced or to produce fish meal and fish oil. So overharvest is a big issue for that one. Other things that we might be concerned about are, we talked about hypoxia and how a lack of oxygen in the environment can affect fish habitat and lead to fish kills. Hypoxic events like that can also increase mortality of benthic organisms, things like clams and other polychaete worms and crabs and things that are really important. So things that are affecting hypoxia, which include warming water temperatures, increased nutrient inputs, other things that are stimulating those algal blooms, can also really have detrimental effects on the forage population. I think those are probably the big things.
Perfect. Thanks so much. I'll let you go ahead on.
Okay, cool. All right, so next I want to focus on sort of the Chesapeake Bay program and what they're doing to address forage issues in the Chesapeake Bay. So the Bay program, a lot of their work is really driven by the Chesapeake Bay Watershed Agreement, which was signed in 2014 by both state and federal entities. And this is basically a formal agreement to restore and protect the Bay. And there's a lot of different outcomes, like 26 outcomes or something like that, that fall under this agreement. And they're all focused on maintaining healthy habitats and a healthy watershed and healthy populations. And that includes forage. So we have this forage outcome in the agreement that states that we will continually improve our understanding of the role of forage in the Bay and develop a strategy for assessing the forage base. So the first part of that is pretty simple to understand. You know, we want to understand better the role of forage in the Bay, which we have a pretty good sense of that based on the information I just presented to you. But developing a strategy for assessing the forage base, what does that mean? And I guess one way to think about it is we really just want to be able to track the availability of prey for key predators in the Bay over time. That's essentially to ensure that we have enough prey available to support those fisheries that we're interested in specifically. So the group that's working on this is the Forage Action Team. They're a work group under the Bay program, and they're a diverse group of scientists and resource managers from around the Bay. And the group was developed in 2014 when that watershed agreement was signed, and they were essentially tasked with focusing on the forage outcome. And the Forage Action Team, they meet a couple of times a year, and we talk about, you know, what's the best way to address this outcome? How can we track forage status? And sort of the one thing that has come out of all of our meetings and our discussions is that the best, most ideal way that we can track forage status is by developing ecological indicators. And those indicators would ideally tell us what the status of forage is in the Bay, like how much prey are available to predators, and how does that change over time. So what are ecological indicators? Essentially, they are tools used to communicate information about ecosystem status and trends. And indicators are ideally based on science, so they take raw data and they transform it into a format that's easily digestible to pretty much anybody. And they should be able to be updated regularly so that you can see how things change over time. So for example, here I have an indicator of aquatic grasses in the Chesapeake Bay, and I pulled this from the Chesapeake Bay report card, which I'll talk about a little bit later. But essentially, this is seagrass abundance survey data sort of translated into a graded status of seagrass abundance from very poor to very good. And you can also see like how the distribution changes across the map as well, which is really cool. So indicators like this are used by policymakers and resource managers and even the public to understand how things are going, maybe for a specific topic of interest, like maybe you're interested in seagrass or water quality or something like that. We can identify where we need to focus our efforts, especially with a distribution map like this. And it's a good way to determine if we're making progress toward our goals or not. And this is essentially what we want to do with Forage. We want to be able to develop sort of a suite of indicators that show the status of forage available as prey over time in a way that's easily digestible and can be used by fisheries managers and policymakers. So there is a scientific process to developing indicators. We really want indicators that are representative of what you're interested in. So in this case, forage. So in our case, the first thing that we needed to do was identify key forage species. So which species are most important prey for predators in the bay. The second thing we need to do is understand the environmental and ecological relationships between maybe some environmental factors and forage species that we're interested in. And this is important because if we don't have really good, like solid abundance data for a given forage species, if we really truly understand the relationship between that species and the environmental factor like water temperature, we could potentially use water temperature as an indicator to sort of estimate good or bad years for that species. And the last thing that we want to do is determine our prey consumption habits. So we need to understand which predators are eating which prey, ideally how much of each, and how that's changing over time. So indicator development can get pretty complicated, but fortunately, a lot of this work was done before I even became coordinator of the Forage Action Team, so that was awesome. And so we have a really good baseline that we can work off of now. So for example, the Chesapeake Bay Program held a scientific workshop in 2014, right after the Forage Action Team and the agreement or forage outcome were established. And this brought together the Forage Action Team and also some other consulting scientists from around the Bay. And they determined the most important forage species within the Bay for some key predators. And the way that they did this was they chose a suite of 5 predators that represent all the different feeding types within the Bay. So they had pelagic predators and demersal. So those are ones that are in the water column versus the ones that are more associated with the bottom habitat. And they also had a variety of piscivores, so those feeding fishes, but also benthivores, so those feeding on benthic species, things like clams and worms and such. And so they had this group of 5 predators. They looked at their gut contents. They used information from it's called the ChesMap survey. It's a fisheries trawl survey that spans the entire Chesapeake Bay, and they also collect gut content data. So they use that information to identify the top prey items for all those species. And this is really where they realized, I think, that benthic invertebrates need to be included when we're talking about important forage in the Bay. So you can see here the key forage listed, bay anchovies #1. We see that's found in most of the stomachs of the species in very high numbers. But then the next 3 are all benthic species. You've got the polychaetes, which are those worms, mysid, which are a bunch of little shrimps, and those little crustaceans that I talked about before too, amphipods and isopods. Yeah, so we found out that those are super important and need to be included and considered in these analyses and these indicators. So what was our next step? Well, so we also needed to understand how environmental factors, things like water temperature, salinity, flow, might affect forage abundance. And so in 2016, one of our Forage Action Team members and some of his collaborators, they conducted this study where they essentially examined a suite of environmental variables and tried to look at their influence on the summer abundance of a whole bunch of different forage species. It included a bunch of fin fishes like bay anchovy. I believe it also had like spot and juvenile croaker, And then they also included benthic invertebrates as well, like polychaetes and a bunch of little crustaceans, things like that. And one of the variables that they looked at was springtime warming, and that's how quickly water temperatures increase in the spring. So for example, well, they essentially they set a temperature threshold, and they looked at how early in the year that threshold was reached. So for example, they might accumulate 500 degrees Celsius by June one year, but maybe they accumulate that in May in another year, and that would suggest that water temperatures warming faster in the spring when it's accumulated by May. And they found that variable, springtime warming, was really a primary driver of forage fishes and benthic invertebrates. A lot of different species reacted to that. They found that with faster warming, so the earlier that temperature heated up in the bay, it led to decreased summer abundance of forage species. And this is important to understand because now we can use this information to develop an indicator based on water temperature warming rates, right? So we can, if we don't have, say we don't have enough bay anchovy data to really talk about how they're doing this year, we could look at how quickly it warmed and be able to say, well, we had a really cool spring this year. We think they probably did okay this year. And ideally, we would be able to make predictions too, because we're looking at information from spring. We can maybe say, well, this is what we think is going to happen in the summer. Another important aspect of this, or outcome, I guess, is that we can get more information about how forage populations might be affected by climate change, which is always an important consideration in the ecosystem work that we're doing, especially in the Bay Program. So another thing that we looked at was habitat. Habitat is an important driver of forage abundance in addition to environmental conditions, because habitats, particularly structured habitats like seagrass beds, oyster reefs, marshes, they are areas where they provide a lot of food for the forage species, and they also act as refuge from predators. And another one of our members of the Forage Action Team conducted this study where they looked at the impact of shoreline hardening on forage abundance. And essentially what they did is they sort of mapped out a shoreline. They looked at what percentage of that shoreline was hardened. So what percentage had like a bulkhead or riprap or something, an unnatural shoreline. And then they looked at the abundance of forage species in those areas. And they found that there's a graph here for croaker as an example. Once they reached a threshold of about, I think it was 10 to 15, they started to see decreases in forage abundance. So at 10 to 15% of the shoreline being hardened, there is a decrease in croaker abundance in those areas. And that, I think they found that the threshold ranged from 10 to 30% depending on the species, but this affected a lot of different species, including blue crabs, I mentioned juvenile croaker, bay anchovy, but some other small forage fishes, things like killiefishes that are pretty reliant on marshy areas. And the reason that, well, at least we hypothesize that the reason that shoreline hardening affects forage abundance is likely due to a loss of natural habitat. So I mentioned that like seagrass and marshes are really important habitats for forage species and for a lot of other species as well. But when you harden those areas, a lot of times you're also disrupting that natural shoreline. So you're maybe getting rid of some marsh or the seagrass beds, and you're therefore taking away some of the refuge that they have, or maybe in their foraging areas. Another thing to think about is a lot of times smaller forage species or little juveniles will use shallow water as a refuge from predators, because larger predators are less likely to go into those shallow waters. You know, they might get stuck on shore or something. So when you put in a hardened shoreline, something like a bulkhead, you're sort of getting rid of that gradation of the shoreline. So they don't really have that shallow water refuge anymore. It's just pretty deep. So those fishes can get in there up to that shoreline and are able to forage effectively in there. So that's what we think is going on. And we're hoping
Mandy, before you go,
yeah.
sorry, I was going to ask a clarifying question because I know you have a couple slides till you get to questions, but could you clarify what you mean when you're using these words like living shorelines or natural shorelines, hardened shorelines? Can you just clarify that a little bit for us?
Yes, so hardened shorelines, so those are what I consider sort of unnatural shorelines. It's where they put in like a bulkhead or riprap or something, like something hardened to really, they use it to stabilize the shoreline. A natural shoreline would be sort of undisturbed, you know, just how you would find it naturally. A living shoreline is something that can be or is, what's a good way to put this? It's implemented. You could create a living shoreline, and it's essentially developing resilience on the shoreline by putting in like stabilizing marsh grasses, or maybe you're putting in an oyster reef in that area, things like that. So it's using sort of the natural ecosystem like habitats that you would find there to sort of help build resilience to the shoreline, as opposed to putting in some of these man-made structures where you put like walls and riprap and things like that. Does that clarify it, at least for you?
Yeah, it definitely does. So I was just typing in the chat. It looks like the top two photos are examples of hardened shorelines, and then the bottom one, it's probably a living shoreline. It looks like there's some Spartina plugs or something in there.
Yes, sorry. Can you guys actually, I was wondering, can you guys see like my mouse when I go across here?
Yes, we can. Thanks for clarifying that.
Yeah, sure. So yeah, these are your hardened shorelines. This would be a living shoreline, as Lee said. They're doing marsh restoration in here and trying to stabilize this shoreline by putting in vegetation. Okay. Where was I? Oh, okay. So we are trying to use this information to develop an indicator as well, since we know that hardened shorelines have an impact on forage abundance. So we're working with the Chesapeake Bay Program's GIS team because they're mapping out shoreline status throughout the entire bay. So they're going through the entire outline of the bay's coast and saying this is hardened shoreline, or maybe you have living shoreline here, or this is undisturbed. And we're hoping to use that map that they have sort of in combination with these thresholds that we developed to identify high-risk areas for forage that are likely suffering from habitat loss due to the shoreline development. Okay, so one of the last things I had talked about was determining prey consumption habits. This work was also done by some previous members of the Forage Action team, Andre Buheister and Ed Hood. They developed these predator-prey consumption profiles. And again, I think I mentioned they used gut content data from the ChesMap trawl survey throughout the bay. And they looked at how the changes in prey consumption for a couple different species changed over time. And I think these graphs are really cool because they show the difference too between different feeding types. So if you look at the top here, we have striped bass feeding, and then at the bottom we have Atlantic croaker and what they're eating. So you can see how striped bass, which is a more pelagic piscivore, is feeding more on these fishes. And then the Atlantic croaker, which is a more demersal or bottom associated benthivore, is feeding more on these polychaetes in particular, but also bivalves and other little crustaceans and things. So it's a really cool way to sort of show differences in the feeding and how it's changing over time. But these, like understanding how this is changing can help us develop indicators that will sort of show the relationship between prey preference and prey availability and how all that changes over time. So the Forage Action team, just this year, we put together an indicator development plan So we knew we wanted to develop indicators. We had kind of gone back and forth over the last couple of years of what those indicators will actually look like. Well, we made a decision this year, wrote it out, so it's official. And we are focusing on sort of 7 priority indicators over the next, I think, 5 years or so. And we've split these indicators up into 3 different tiers. The first tier is sort of the most simple indicator that we can develop, and that is essentially just a time series of actual forage abundance. And this will be good for ones where we actually have data. So for benthic invertebrates, we have really good data from Chesapeake Bay long-term monitoring program. So we can track how polychaetes have changed over time. We can do the same thing with some different demersal fishes and pelagic fishes as well. But then again, as I mentioned, we have some species that are a little more difficult to track. So we have a tier 2 where we're looking at the relationship between these habitat and other environmental factors and how they affect forage, and then creating indicators out of that. So I talked about the springtime warming indicator and hardened shorelines. We also have a couple other studies that NCBO is funding focused on habitat suitability indices, which essentially they're doing a lot of modeling and quantifying the relationships between a lot of different environmental variables and forage species, and figuring out which ones are the most important variables that affect forage abundance, and showing how, as those variables change over time and space, the distribution and abundance of forage species in Chesapeake Bay also changes over time and space. So that's sort of something else that we have coming down the pipeline soon too. And then the third tier is sort of the most complicated tier we have. That's the predator consumption. So those diet profiles that I showed, those really colorful graphs of how consumption changes over time. That's a little more complicated. You need a lot more data. You need to have sufficient predator data and sufficient prey data to be able to do that. So that's something that will probably be a little bit further down the pipeline, but something that we've obviously started doing that with the other studies that I talked about before. All right, so we have a plan for these indicators. So how exactly can they be used to inform management? And I mentioned at the beginning, we really hope that these indicators will inform both habitat and fisheries management. From a habitat aspect of it, if we can identify the shoreline development trends throughout the bay, since we know that that shoreline hardening has effects on forage abundance, we can use that information to help target restoration and conservation efforts throughout the bay in order to help maintain habitat for the forage base. And that might look like informing regulations for hardened and living shorelines. I also mentioned that some of these other structured habitats that are, kind of key to the bay in general, things like marshes and SAD and oyster reefs, those are also important for forage abundance. So knowing how those habitats can affect forage species and forage production, we can also prioritize those habitats for conservation and restoration efforts as well. And additionally, understanding how sort of water quality and those other environmental conditions affect forage, things like nutrient pollution, how that affects hypoxia, as well as like freshwater flow and salinity and water temperature. Those all impact forage in some way. And those are kind of tied into what we call TMDLs, the total maximum daily load. So, and that's also a water quality management effort. So we can also ideally inform something like that as well. Sort of the big thing though is fisheries management. And I guess the easiest way to think about it is informing the reduction fishery. So being able to understand how forage abundance is changing over time in order to develop targets or I guess harvest targets for the reduction fishery. that can help people make management decisions. But another way to think about it is ecosystem-based fisheries management. So how can we effectively manage the forage populations to manage the other fishery species that we're interested in, things like striped bass and blue crabs. So all these things are really tied together. And you know, this is, the end goal is really being able to take actionable or make indicators where management or managers can make actionable decisions. So I'll stop there again quickly for questions before we move on to the next segment.
Yeah, so Mandy, we had, we got like about a little less than 15 minutes left, just as a heads up on the timing thing. Yeah, so maybe just we'll ask one question, but so Minister Bryan was asking about his experience like fishing up in New Jersey and Raritan Bay versus, you know, kind of the Chesapeake Bay, and like, why do these places that are so close, like, they seem to look kind of different in terms of the forage there. And he actually made a point of like the alewife are off the hook. You know, a lot of alewife 10 to 12 inches swimming around in Raritan Bay on a regular basis. And that doesn't look so much like that in the Chesapeake right now.
I just want to clarify, the forage base looks different in like up in, I think you said Narragansett Bay?
Raritan Bay, so up New Jersey.
Oh, okay. Yeah, why do they look so different?
Yeah. Yes.
Okay. Yeah, I mean, a lot of it is geography. I don't know a whole lot about Jersey forage populations or anything, but I imagine a lot of it has to do with just the geography and the different environmental conditions in that area, and also probably partially sort of the Chesapeake Bay is super agricultural, and all that nutrient input from the farmlands and things and the development along the Chesapeake Bay shoreline. I'm sure that that's having a huge impact on those like riverine species like alewives, shad, and other things that are really important forage. But yeah, no, it's a good question, but I can't speak to any specific example, unfortunately, since I'm only really versed in Chesapeake Bay. But yeah, just different environmental conditions and probably difference in the community structure as well. Maybe different predators that are out there.
Oh, cool. There's well, I guess like just in the interest of time, maybe I'll hold off on my follow-up question, but maybe I'll ask it at the end if there is time.
Okay.
Cool. Thanks, man.
Yeah, I'll rush through these quick. We're actually at the final stage. I'm presenting now just some potential resources. I was going to click through some of these, but maybe I won't do that now. So one of the things I wanted to provide information about are some of these other Chesapeake Bay indicators. You can access these. I mentioned previously the seagrass indicator that I pulled up in my presentation is from the Chesapeake Bay report card. This is developed by University of Maryland Center for Environmental Science and their Integration and Application Network. So you can go in there, click that link, and it will have a whole bunch of indicators ranging from different variables of water quality. They also have some living resource indicators like bay anchovy and benthic abundance and how that changes over time. And they also have like those really cool distribution maps that I think are super neat to look at. So there's that. And then there's also Chesapeake Bay or Chesapeake Progress, which is where we hope the forage indicators will live when they're completed. That website also, it puts those indicators in the context of the Watershed Agreement outcomes. So you can read about those. And then those indicators are cool too because, or the website is cool because for each indicator, you can also download data. Like for example, if you look at the Chesapeake Bay blue crab trends, you can see the graph there, but then they also have a button where you can download an Excel file of the data. So that's a good way to get some just basic information and data if you want to do some data analysis. Super quick. I wanted to talk briefly about my graduate research because it actually informed this lesson plan that I created. I was a GK-12 fellow in grad school. So I was essentially an assistant scientist in the classroom where I created lesson plans. And so my research was focused on, like I mentioned at the very beginning, juvenile blue crab survival and identifying key predators. And the way that I did that was I did what are called tethering experiments. This is gonna sound so brutal. So what I did was I super glued these little tiny juvenile blue crabs to fishing line and staked them out in seagrass beds. And I put GoPros around them, and then that way I could capture images of what was feeding on juvenile blue crabs. So here we have a northern puffer eating one and an adult blue crab. Saw a lot of cannibalism in my studies. Another aspect of that, to get a little broader sense of what was eating things in terms of fish, I did some gill netting. And so I collected a lot of different fishes and did gut contents to see what was eating blue crabs. So here's a striped bass that I caught. Here's us collecting some fish from the gill nets. So essentially my research was sort of motivated by this decline in the blue crab population in 2012. And one of our hypotheses for that decline was that maybe there was just increased predation on juvenile blue crabs that summer, because we also had anecdotal accounts from watermen saying that they were seeing tons of juvenile blue crabs in the stomachs of red drum. So I created a lesson plan for middle school life science class to sort of demonstrate the predator-prey interactions and use that sort of story of declining blue crabs as the background for this project. And basically I made little Play-Doh stomachs of predator fish species. And then inside they had different bead types that represented different prey types, like crabs or fish or plankton. And so the students would have to dissect their stomach, identify what kind of prey they had in there. And then they would learn how to use triple beam balance and weigh out their different predator types and graph them. And then the class would come together and pool all their data, and then they would determine which predator species was most likely to impact the blue crab population. So that was just a lesson that I developed based on my research. I think it's relevant here. Juvenile blue crabs are key forage, but you can also sort of modify this lesson to identify key forage species for predators and talk about decreasing or how changes in forage and predator populations can affect each other. Last thing, so there's a link here to the PDF of that lesson plan that I developed. VIMS also has an education, VIMS is Virginia Institute of Marine Science. So they have this education link where you can go in. If you go in here, you can find Virginia Science and Educators Alliance. That's where all the other lesson plans are, as well as mine from that GK-12 program. There's also a link in there where you can go to Bridge, I believe it's called, and they have a lot of sort of NOAA-backed lesson plans that you can use in the classroom as well. The very last thing I want to talk about, which I'm going to pull up since it is a little more complicated, is the CHESMAP survey, which I talked about. This is the fisheries trawl survey that's bay-wide. This is a really great resource to get some data. So you can come over here to species data. You can select a specific species if you want. It gives you a lot of background information on it. But you can also look at these really cool distribution maps of where they're catching a given species and how much and how that changes over time. I hope this, okay, here we go. So we can look at, say 2019, so last year, where were they catching species, and then at what abundance were they catching them. So this is really cool. You can look at how things change in space and time. And then if you want the actual data, you can request that from VIMS. You just have to go to interact with our data. And then they will have a request form I won't go through that because you have to sort of log in, but you can get abundance indices for specific species in there. And you can also request the gut content information, either based on predator or prey diets. So this is what it looks like. You can go in and say, I'm interested in striped bass, what's eating, or what do striped bass eat? And it'll spit all that information out. Or you can go in and say, I'm interested in this specific prey type, like what eats bay anchovy, and it'll spit out all that information. But yeah, you have to register and just ask for data and they will give it to you. And I think that's it. Let me get back here. All right, questions? Sorry for the rush at the end there.
No, that was perfect. Thank you so much, Mandy. Yeah, it was really super interesting presentation. And I really like that this topic gets into the complexity of the systems at play, and it highlights some of those things that as educators are really important for us to make sure come through, the cross-cutting concepts like cause and effect, stability and change patterns, all that kind of good stuff. And then I just wanted to point out like that MWEE connection. So I was hearing some really great opportunities for driving questions in a lot of the information that you shared. So how do stream temperatures impact the prevalence of forage, or how do shorelines, whether they're hardened or natural, potentially impact forage also. So I think there's just a lot of great opportunities for inquiry here, and really appreciate you bringing that to bear. And then of course there's the management and policy connection as well. And we know through the MWEE students engage in stewardship and civic action, and there's I think a lot of potential ideas there. So thank you so much for sharing. There's a couple questions in the chat. And shout out to Amanda, who was in your GK12 cohort, who's also mentioning the resources. That's great.
I saw
we have a question about youth initiatives and how like where you see an opportunity for students to kind of step into that process. Somebody's mentioning that they're currently trying to teach youth about how to raise forage fish. Any like insights there?
How to raise forage fish?
So I'm not sure, Minister Brian, if you are interested in coming off mute, you're welcome to do a quick explanation of, or maybe elaboration on your question.
I'm sorry. Thank you so much. We're currently raising forage fish. They're raising bluegills to go back into some salt marshes. But I wanted to know what other youth initiatives could we implement that could help the process, whether it was just planting water grasses, or what else would you suggest?
So it sounds like what other sorts of actions can students engage in to kind of support forage? So I think the habitat piece seems like a critical one.
Yeah, I was gonna say even that suggestion you gave is a great one. Yeah, it's just ensuring that we're restoring the appropriate habitat types. For bluegill, I'm not super sure or where you're doing that work. But yeah, if you know what kind of habitat type would be suitable for bluegill, ensuring that you're restoring and conserving that habitat type, that would be good. Yeah, that's probably the easiest one, I think, to get into, especially for kids doing that kind of thing.
Yeah, that's great. The other thing that comes to mind is, you know, thinking about like what are the root causes of some of the challenges that that we face. So, you know, if we're talking about increased stream temperatures, for example, that might be linked to climate change. So, like, you know, what are the root causes there, and what are the potential interventions that you could take across that process, depending on the grade level and the understanding that your students have about that whole process. But Bart, are we going to chime in?
I was, but I'm seeing the time, so I don't know if I want to go.
I just want to add one thing to what you said, Elise. That's a good point. With stream temperature, we know forest buffers tend to reduce stream temperatures and keep them cooler, which is often ideal for a lot of these fishes. So maybe just planting trees along a bank and sort of helping restore those riparian buffers would be a good one.
That's a great other suggestion. I do see that it is 4:31. I see a couple folks dropping off, so I welcome anybody to stay on. I think Mandy might have a minute or two to answer questions. And if you'd like to chat with her afterwards, we could potentially connect you all over email. But thanks everyone for joining us. We hope that to see you at our next webinar, which is again on December 1st, and we'll send out that email. If you're not already on our list, feel free to drop your email into the chat and we can add you to that. So thank you. And for anyone who wants to stay on, feel free to unmute at this point, and you can ask a couple more questions if that's okay with you, Mandy.
Yeah, that's fine.
So,
I'm gonna stop sharing my screen. Oh.